review video that I made for my biochemistry class, so I thought I'd share it with you too in case it could help. It covers intermolecular forces, pH, pKa, all of that stuff, PI, amino acids, levels of protein structure, protein purification, and a little bit on binding affinity. So hope it helps. Some of it is going to be obviously specific to my class, but I also will post links to an outline as well as the slides and hope something of this can help you. Key to really understanding biochemistry is not to memorize. So memorization might get you through your MCATs, but it's not going to make you a good biochemist, and it's not going to make you a good doctor. Instead, it's really important to understand these concepts, be able to apply them to new situations, be able to see how biochemistry really is life, how what we're learning in class really does affect what's happening in our bodies, what's happening around the world, and how we can understand things through the lens of biochemistry. So that's what I really care that you guys can do, not that you can memorize things. So I really want you to know these key concepts and be able to apply them. In order to be able to kind of think molecularly, we need to be able to recognize our functional groups. So these are the functional groups that you're going to be held responsible for. We should know our hydroxyl, our amine, our amide. So remember that an amine is going to have a nitrogen with hydrogens. With an amide, you have it next to a carbonyl carbon. So an amide and an amine are two separate things. Another common confusion is a sulfhydryl group or a thiol group. This is going to be an SH. So when you see sulf, when you see thiol, when you see mercapto, those are all hints that something has a sulfur. Because sulfur is right under oxygen on the periodic table, you're going to see sulfur analogs or versions of things that would normally have oxygen. So we have a hydroxyl, which is an OH, and we have a thiol, which is going to be a sulfur version of that, so an SH. So cysteine has a thiol group, but methionine does not. It's not enough to have a sulfur. To have an actual sulfhydryl, to have a thiol group, you need to have the SH. We also need to be able to recognize an ester and an ether. And so remember how we can have sulfur versions of things? Well, we can have a thioether, which is going to be a sulfur version of this ether. And that's what we find in methionine. Remember our carbonyl, our C double bond O, which we can find in various contexts, including in a ketone where we'd have a carbon on either side, or an aldehyde when we have a hydrogen on one side. Then we've got our ester, we've got our carboxylic acid, and we have our amide. And I want you guys to be able to know what interactions these could form as well. So let's talk about those interactions. Remember that we can kind of simplify things down and think of all these intermolecular forces as coming down to electrostatics, as coming down to charge-based attractions. We can talk about full charge attractions, in which case we're talking about ionic bonds, which remember we can call these salt bridges, or we could be talking about partial charge attractions. And those could be permanent partial charges in the case of a molecule with a permanent dipole, or they can be temporary dipoles. They could be temporary charges. No matter what the source of this charge, and we'll talk more about those various sources, but we can kind of think about them as being charge-based, and therefore we can think about them using Coulomb's law. And so you're not going to have to do any sort of calculations or even recognize things in this formula. This just kind of shows you how these all combine. So what's going to happen is that the force is going to depend on three things: the strength of the charges, the distance between them, and the polarity of the intervening medium. So the strength of the charges. The higher the charge, assuming we're talking opposite charges here, the more the attraction. We have Romeo and Juliet yelling at each other that they love each other. They're going to feel the love very greatly. But if they're far away from each other, then they're going to have a harder time feeling their attraction, hard time hearing that love. And so the same for molecules. The bigger the distance between two molecules, the weaker the force attraction is going to be. The shorter the distance, the more the attraction. And the distance is going to have a bigger effect. Basically, the force is going to die off more quickly for the weaker interactions than for the ionic bonds. But both of them are going to fall off with distance. Now, the third thing is the polarity of the intervening medium. So this is going to be our dielectric constant, also known as relative permittivity. What's really important is that this is of the intervening medium. So this is like your solvent. This is not going to be the polarity of the charged molecules. Instead, it's the polarity of the solvent. Or basically, you can think about Romeo and Juliet. If they're yelling louder, stronger charge, more attraction. If they're farther away on a football field, across the football field, weaker attraction. And if that football field is filled with football players running and distracting them, well, they're going to have a harder time feeling each other as well. Similarly, in a solvent that is higher polarity with a higher dielectric, something like water versus something like lipid, well, now what's going to happen is those forces are going to have a harder time feeling one another. So if we have something like a salt bridge in the center of a protein where it's all hydrophobic, it's kind of like a low polarity environment, that's going to have a stronger attraction, be more significant, stronger force than you would at, say, the surface of a protein where you're in this watery aqueous environment. And the same would hold true for things like hydrogen bonds and all of our IMFs. Basically, the charge, anything which when we're talking about charges feeling one another, it's going to be easier to feel one another in a lower polarity solvent or a lower polarity environment, such as if in the center of a protein. So now let's talk about the different types of IMFs. So the strongest ones are going to be our ionic bonds. And when I talk about strongest here, basically thinking about all else being even, thinking about the same distance and that sort of thing, the same solvent that you're in. The strongest IMF is going to be our ion-ion, which is going to be our ionic bond, our salt bridge. And we're often going to be talking about salt bridges in the context of the amino acids and various regions on the protein forming these salt bridges. They're going to be the strongest because they have those full charges. And remember, the bigger the charge, the more the attraction. Now we're going to have various temporary or various permanent dipoles. But next, we're going to have our permanent dipole type attractions, the strongest form of which is going to be our hydrogen bond. And so we consider the hydrogen bonds as kind of like their own separate class, and we'll discuss them in more detail in a minute. But basically, when we talk about, we can get this partial charge by having a polar molecule in a polar molecule. To be polar, something is basically you have an electronegative atom connected to something that's less electronegative, and therefore the electrons that they're sharing in their covalent bond, the electronegative atom is going to hog them, leading it to be partly negative and the thing it's attached to be partly positive. When we talk about electronegative atoms in the context of biochemistry, we're typically dealing with our oxygen and our nitrogen, with oxygen being the real electronegativity queen. And so when oxygen is, say, attached to a hydrogen, that poor little hydrogen with its single proton, it has it's no chance at like pulling in any electrons. The oxygen is really just going to hog them. And so water is a great example of a highly polar molecule where the oxygen is going to be partly negative and the hydrogens are going to be partly positive. And that's going to be a permanent dipole because the oxygen is always going to be more electronegative than the hydrogen. So you're always going to have a dipole. But you can even get a dipole in molecules that don't normally have a dipole. And so this can happen if something charged comes near. And so remember that when we have a non-polar molecule, say, so something without a dipole, something that maybe it's the hydrocarbon, the electrons and the hydrogens and the carbons, they share electron pretty fairly. So you don't have any sort of dipole. But those electrons, they're kind of like moving all around randomly. And so what's going to happen is that if something charged comes near, well, now the electrons that are on that side of the molecule, they're going to either get attracted or repelled. So if you have a negative charge come near, well, now those electrons are going to flee from it. They're going to get repelled, and then the electrons in the molecule next to them are going to get repelled, and then the next and next and next, and you get this sort of chain reaction. So that can happen if an ion comes near. It can happen if a polar molecule comes near. And it can even happen randomly. So if those electrons randomly moving around just happen to be more on one side than the other, this can set off a chain reaction. When we talk about like these transient dipole-based attractions, we often refer to these as like van der Waals forces. When we talk about those like random events where just like this randomly happened to be somewhere, we typically refer to those as London dispersion forces. We use this term van der Waals fairly broadly, but so whatever of these terms you want to use, we sometimes talk about when we talk about these really weak forces, we talk about like nonpolar interactions, all that stuff. But basically, so know that we have our ionic bonds, our salt bridges, our hydrogen bonds, our other permanent dipole, and then our transient dipole-based attractions, our London dispersion forces, and other things like that. Now, all of these, even our strongest ones, even our ionic bonds, these are going to be individually weak. And there's no way that a single bond can hold together a whole protein. But really, when we put a bunch of them together, well, now we have strength. And so we can talk about the relative strength. We can talk about a hydrogen bond being stronger than like a dispersion force, but still these are going to be individually weak, but collectively strong. So remember the example of the phone book where you interlace the pages of the phone book and the friction adds up enough to pull a truck. That we can get that sort of interaction in our molecules as well. So you should be able to predict the types of IMFs that various molecules can form with one another and with the solvent. And this includes the solvent. So we need to think a lot about the solvent in biochemistry. And so our solvent is almost always going to be water, unless we're talking about like in a lipid environment in the center of a protein or something. We're going to be dealing with water. And remember how water is super duper polar. Water really loves other water. So basically, remember that it's got that partial positive, partial negative, and now the water molecule next to it has a partial positive and a partial negative. They're like born for one another. And so water is really going to want to hang out with other water molecules. And it's going to want to form hydrogen bonds, which we'll discuss more in a minute. But water is really happy hanging out with other water molecules. Now, say you stick something else in there. Well, now water has to make a choice. Is it gonna give up its water interactions to like interact with this new thing? Well, it's going to depend if that new thing is going to offer something better than the water molecules, or at least as good. So if you can form other sorts of like polar interactions, if you could form interactions with an ion, all this stuff, water's like, okay, I'll swap one of my bonds to my buddy to form an interaction with you. But if not, then I'm just gonna like kind of clump you together and maximize my interactions with my water friends. And so by maximizing my interactions with my water friends, I kind of gang up around you and I'm not going to dissolve you, or if you were in the solution, I'm going to clump you all together. And so we'll discuss that more in a minute in the hydrophobic effect. But if we want to kind of like predict whether something will be water soluble, we want to look for things that are going to be more polar and or ionic molecules. And remember, it's not enough to have a single nitrogen or a single oxygen to be polar. We kind of need to consider the overall molecule. So if you have a mostly nonpolar molecule and there's like one nitrogen, such as in like tryptophan, we'd still consider that to be nonpolar. Even though it does have like one of those nitrogens. But if we look and we see a bunch of nitrogens or a bunch of oxygens, those things are typically going to be hydrophilic and water soluble. Whereas if you see basically like a pure hydrocarbon, that's going to be hydrophobic. And remember, phobic, it's not afraid of the water, it's just avoided and excluded by that water. But it might be happy in fats. So that's why we can talk about like fat and water soluble vitamins. So binding affinity as well as like solubility. So remember that solubility really just involves like bonding interactions between our solvent and our solute, between our solute and our solute, between our solvent and our solvent, which is going to be preferred is ultimately going to come down to thermodynamics. So remember, we want that negative delta G. We want that change in free energy to be negative. We want our products to be happier than our reactants. We want the bound form to be happier than the unbound, or the previous state, the new state to be happier than the old state. We want, when we're at that bottom of when we're at the bottom of the roller coaster, we're a lot more relaxed than at the top. And similarly, when molecules have a negative delta G, that's telling us that they're kind of like more relaxed. They're happier in their new form. And so how do you get this? Well, this can come from having a decrease in enthalpy, so our delta H, forming more favorable bonds and or interactions, and or an increase in entropy, so giving molecules more freedom to move about randomly. So there are a couple of key points to think about when considering these terms. One is that when we talk about enthalpy, this is not just referring to covalent bonds. It's also referring to like interactions. So we can think about enthalpy as involving not just the any sort of covalent bonds being made or broken, which we're not going to see when we just dissolve something. But we are going to have changes in enthalpy because we're changing the interactions that we have. We're switching up some of those solvent-solvent interactions, solute-solute, solvent-solute. So you're having a change in these interactions. And if you make things more favorable, well, now you're going to get a favorable change in enthalpy. The other thing to keep in mind is that we need to consider all the molecules in our environment. So we're not thinking about, if we're thinking about two molecules binding, or if we're thinking about a molecule binding one other water molecule, that's not, it can't be happening in isolation. It's happening in this environment, this aqueous environment. So we need to consider not just those two molecules, but also all the water around it, all the whatever molecules are around it. We need to consider those as well. So we need to kind of take into account the system rather than the individual molecules as if they were isolated. And so when we talk about the entropy, we need to think about the entropy of our solvent. We need to think about the entropy of water. Because what's going to happen is, yes, if you take two molecules together and you stick them together, well, now, yes, you've decreased the entropy of those two individual molecules, but they're not in isolation. And so all those water molecules that were around them, well, now you've released those. You've increased the entropy of the water by pushing those things together. So this is why you can have things bind and still have an increase in entropy. So it sounds really counterintuitive if we're thinking entropy, randomness, disorder, but it's because you have to think about the water as well. So going to that water, let's talk about the entropy of water, because this is going to be the driving force for things like protein folding. It's going to be this hydrophobic effect. So remember that hydrophobic molecules are not intrinsically attracted to other hydrophobic molecules. So we talked about like ions being attracted, like cations being attracted to anions, and we talked about polar molecules being attracted to other polar molecules. But we cannot say that hydrophobic molecules are intrinsically attracted to other hydrophobic molecules. Instead, they don't really like one another, but they're kind of just forced together. And they're forced together because other things don't like them. More specifically, the water around them does not like them. They offer nothing for that water. Remember that hydrophobic molecules, they're like hydrocarbons, things that don't have any sort of like permanent polarity. The only thing that they can do is those like transient weak forces. Yeah, water's like, no, I want to hang out with other water molecules. I want to hang out with charged things. They have all this water around me. Why should I hang out around you? But if you stick something in water, well, water's going to have to kind of cope with it somehow. And so water's going to try to maximize its water-water interactions. And the way that it can do this is by kind of like clinching around a nonpolar thing. This is going to take the water out of commission around that thing. If you think about the free water in like the bulk water, like not around something like nonpolar, but just the water in the solution. So remember those hydrogen bonds, they're individually weak, collectively strong. If those waters are constantly making and breaking interactions, but if they break one, well, there's another water molecule right next door. So they can kind of like dance around and be moving around happily, going from one water to another water to another water. But what's going to happen is that in order to maximize their interactions around something nonpolar, they're kind of stuck more in place. So you get this solvation shell. You get this like clathrate water, this water that is going to be kind of like constricted. So that water has a low entropy. But now if you kind of reduce the surface area that it has to cover, you're going to take less water molecules out of commission. And that's going to free those water molecules that were tied up, giving you an increase in entropy. So by pushing together the nonpolar things, by folding up a protein so that you put those nonpolar parts together, those hydrophobic parts together, well, now you're going to be able to increase the water's entropy, and this can provide a very, very strong force for binding, for folding, for all of these things. The hydrophobic effect really comes down to the entropy of the water. This does not come from any intrinsic force, but any of attraction. But basically what happens is that once those hydrophobic things are pushed together, well, now you can have those transient dispersion forces come into play. So remember that those forces are going to only be kind of like work at a very, very close level. But when you push all distance, but when you push all those molecules together, well, now they're really close to one another. And if they're like kind of like held there where they can't escape, well, now you can get these kind of transient interactions forming these strong, being collectively strong. But that's not the driving force of the hydrophobic effect. Instead, it's just kind of like a consequence that happens after those things are pushed together. The driving force is going to be that entropy. Okay, so let's talk more about what kind of bonds those water molecules are forming with one another. They're forming those hydrogen bonds, those H bonds. And remember that those are going to be our strongest type of dipole-dipole interactions. And basically, they require a couple different things. They require a donor, which is a hydrogen on something electronegative, so an oxygen or a nitrogen, sometimes a sulfur, which is why we consider cysteine to be like polar. Basically, sulfur is a little like, it's not very electronegative, but it's more so, it's still able to kind of like form these hydrogen bonds. So we can consider that hydrogen on the sulfur can contribute as a hydrogen as a donor, and that the sulfur itself, it can act as an acceptor, because basically what's going to happen is it has a lone pair of electrons, and when you have an electronegative atom with a lone pair of electrons, well, now it can serve as a hydrogen bond acceptor. But again, sulfur is kind of weird for this. Normally what you're seeing is you're going to be seeing an oxygen or a nitrogen. We're going to be looking for things like an carbonyl or an amide. Remember that they need that lone pair. So if that lone pair isn't available, if that lone pair is tied up in resonance, then you're not going to be able to act as an acceptor, even if you could still contribute to the polarity of a molecule. So like all those IMFs, even though we think about hydrogen bonds as kind of like being stronger than other IMFs, other than ionic ones, basically it's still going to be individually weak, collectively strong. And it's going to be stronger than those other ones because of a few things. One is that they have a short distance, so that hydrogen's really small, remember? So they can get really close. And we talked about how the closer they are, the stronger the force is going to be felt. We have Romeo and Juliet in an elevator versus on a football field. So we're going to have a stronger attraction. Also, because hydrogen is such a wimpy, like polar when it comes to electrons, you're typically going to get a large dipole. So if you have an oxygen attached to a carbon, yeah, the oxygen is going to pull in the electrons, but it's not like if it's attached to a hydrogen where it really gets a good grab on them. So you're going to get a big dipole when you have one of these hydrogen bonds, donors and acceptors typically, like, and the bigger the dipole of the individual molecules, the stronger the force is going to be. Also, these kind of like have a partial covalent character, which you don't need to worry about. But basically, because you kind of have things that are so close together, you can have overlap and things like this. And to be most effective, to have maximal strength, you need to have a specific orientation of the donors and of the acceptors. And so this is part of the reason why, in addition to steric hindrance, protein secondary structures take distinct forms, as we will review in a minute. But you should be able to identify potential donors and acceptors and draw dotted lines for potential hydrogen bonds, both between the molecules and typically between the molecules and the solvent, which is almost always going to be water, especially when we're talking about hydrogen bonds. Okay, so another thing about water, we can talk about acids, bases, pH, pKa, pi, and buffers. Sounds like a lot, but really there's this few key terms that you need to know, and their relationship to the pH. So the pH, this is our measure of free proton availability. This is a solution level thing. It's going to come from the effect of all acids and bases in a solution. So when we're talking about the pH, this is going to be the solution level. But your solution might have a bunch of different acids in them. And for each of those acids, for each acid site, we have a pKa, which is going to tell you about the acid's strength. So that's going to be the individual site. Then we have the pi, which is going to be telling us about the molecular level, the molecule level. So we can have a molecule with multiple acid sites, such as a protein, and this is going to have then a pi. And then we can talk about a pKr, which is just going to be the pKa of an amino acid side chain or R group. Now remember, not all of them are going to have one, but some will. Okay, so going back to our pH. The pH is a measure of free proton availability. And remember, this is the solution level, so it's going to come from the contributions of all the acids and bases in a solution. So the more acids there are, the more you're going to be having things donating protons. So you're going to be more acidic. If you're more acidic, you have a lower pH because it's an inverse log. If you got more bases around, they're going to be taking up those protons. So there's going to be fewer free protons available. You're going to have a higher pH, more basic solution. So the more protons there are, the lower the pH, and the more likely things are to be protonated. So no matter what we're talking about, at a lower pH, things are more likely to be protonated than they would be at a higher pH. But how likely something is to actually be protonated will depend on how much it wants a proton, which is going to be reflected in the pKa. So imagine a lottery ticket, and the lottery has a really bad chance of, you have a lottery where there's really, really poor odds of winning. So each of the tickets has a really, really low probability of winning. Well, now you need a lot of the tickets in order to win the lottery. Similarly, if you have a stronger acid, what it's going to have is basically you're going to need a lot more protons around in order to find like half of it be in its protonated state. And so what this means is that you're going to need to get to a lower pH in order to protonate it. Whereas if you had a lottery with a good odds of winning, you don't need as many tickets. And so this would be like a weaker acid, a stronger base. Basically, you need fewer protons around for it to be protonated, and therefore, even at a higher pH, it will still be protonated. And so basically, we have this measurement that we can say, okay, when it's 50-50 acid to base, well, then we'll call this pH the pKa. So the pKa is just the pH at which half the copies of an acidic site are deprotonated. And so remember, that's our conjugate base, which we can abbreviate A minus, and half are in the protonated state. So that would be our conjugate acid, which we can abbreviate HA. But remember that our HA, this can actually be positively charged and the base can be neutral, or the base can be negatively charged and the acid can be neutral. So the pKa doesn't directly tell us about charge, but it does tell us about the ratio of protonated to deprotonated. Remember that at a lower pH, you're going to have more protons available, things are more likely to be protonated. And if we said that the pKa was the halfway mark, well, then if we're at a pH that's below that, we're going to be predominantly in the protonated acidic conjugate acid form. Whereas if we're at a higher pH than the pKa, there are fewer protons around, so we're more likely to be in the conjugate base form, the A minus form. But remember, it might be negative or it might be neutral. And remember that basically for every pH unit change, you get a 10-fold increase in the amount of protons or decrease. So if you go up 1 pH unit, then you're going to basically go 10 times less protons available. If you go down 1 pH unit, then you're going to have 10 times more protons available. Similarly, if we go 1 pH unit away from our pKa, if we go 1 higher, we're going to have 10-fold more of our conjugate base. And if we go 1-fold lower, we're going to have 1 pH unit lower, we're going to have 10-fold more of our conjugate acid. If you go 2 away, well, now you've got 100-fold difference, 3 away, 1,000-fold difference. And basically, you can think of that all as basically almost all going to be in that one form. So we can use the Henderson-Hasselbalch equation if you need actual numbers. I'm not going to require you to do any calculations with Henderson-Hasselbalch, but I do want you to recognize that, like, if something is a couple pH units away from its pKa, it's all basically always going to be one form. So if you're at a lower pH, then you're going to be almost all in the protonated form. And if you're at a higher pH, you're going to be almost all in the deprotonated form. But when you're in that middle, when you're at that at or near the pKa, well, now you have relatively equal amounts of each. And the significance of this is that you have a good buffer. So a buffer is just a solution where you have relatively equal amounts of conjugate acid and conjugate base. And therefore, you have enough acid to counteract any added base and enough base to counteract any added acid. So you're able to maintain a fairly consistent pH. We can see this visually if we draw a titration curve, and you'll see that the flattish regions around the pK, there's a flattish region around each of those pKa's, which is going to correspond to that buffering region. If we want to choose a buffer, we want to choose one that has a pKa that's close to the pH that we want to be held at. Once we go like about one plus or minus 1 from the pH, from our pKa, well, now what's going to happen is we're going to have tenfold more of one than the other. And if we go a little farther away, we're going to quickly have not enough of the one to counteract the other. And so what's going to happen is that we can define a buffering region as basically about 1 pH unit above or below the pKa. And so if you're choosing a buffer, one of the most important things to consider is whether you want one with a pKa that's near the pH that you want. And then there are other considerations, such as whether it's compatible with other things in your mixture as well. So that was our pKa. And remember, the pKa was telling us about protonation state, but not about charge. The pi isoelectric point, it is telling you about charge. So electric, it's telling you about charge. And so the pi is going to be the pH at which a molecule is net neutral. And so remember, we're talking about the molecule level. So this can be made up of multiple different acid sites. Each of those sites would have a pKa, and then the molecule overall would have a pi. So here we are talking about charge. So at a pH that's below the pi, a molecule is positively charged. And at a pH above the pi, a molecule is negatively charged. And it's going to come from the combination of acid-base groups in a molecule. And we're most commonly going to be talking about pi when we're talking about proteins, which, remember, are going to have various combinations of acidic sites. For all of these, though, we can just remember that at a lower pH, things are more likely to be protonated. And if things are more likely to be protonated, then things are more likely to be positive. And so if we're thinking in terms of where the neutral point is, then below that, you're more likely to be positive. If we're thinking about where the protonation state is, below that, you're more likely to be protonated, but you may or may not be positive. But when we're talking about isoelectric point, we're talking about charge at the molecular level. When we're talking about pKa, we're talking about protonation at the single site level. And when we're talking about pKr, well, now we're just talking about the pKa of an amino acid R group or side chain. So remember that only some of these amino acids will have a pKr, and we can refer to these as acidic and basic, as we'll review in a second. But all amino acids are going to have a C terminus, and they're going to have an N terminus, each of which can act as an acid or a base. Now, the C terminus is going to basically our carboxylic acid group. That has a pKa of around 2. So I want you to know that value. You have a pKa of around 2. What this means is that when we're at a pH of around 7.4, we're going to almost entirely be in the deprotonated state, our carboxylate state. And our amino terminus, our N terminus, it has a pKa of around 9 or 10. So we're going to be at least 2 pH units away from the pKa. And so we're going to be almost all in the protonated form. And so what's going to happen is that we find the molecule predominantly in its Zwitter ionic form. So remember, Zwitter ion, it has a negative and a positive charge. that cancel out. And so this would be this zwitterionic form. And I always want to see you draw amino acids with their zwitterionic form. I never want to see you draw in the non-ionic form. Remember that you're never going to have this amino group deprotonated and this the carboxyl group protonated. No matter how many times you see it in a textbook, you're not going to see it in the body or in reality, because you're going to have deprotonated one on one side and protonated one on the other side. Okay, so yeah, so I don't need you to memorize pKrs. I do want you to know those relative those approximate pKa's for the N and C termini, as well as know for the amino acids that have a pKr, what the predominant form of each would be as you would find it in the body. So remember, that's the ones that I care that you know about in terms of acidic versus basic. We're going to talk about the basic amino acids as being arginine, lysine, and histidine. And we call them basic because in their neutral form, they act as bases. Remember that when I talk about neutral form, I'm talking about neutral in terms of that molecule, that R group being neutral as an uncharged. I'm not talking about neutral pH. So if I'm talking about pH, I'll say the pH, like a neutral pH, or just give you a number. But I'm not, when I'm talking about neutral in terms of a molecule, I'm talking about the molecule being neutral, the molecule being uncharged. If we look at histidine, lysine, and arginine, well, when they are uncharged, well, now what can they do? Now they have an amine group that can act as a base, take a proton. Protons are positively charged, so if they take a proton, if they act as a base, well, now they become positively charged. And so we call these basic amino acids because in their neutral form, they acted as a base. And remember that once they act as a base, well, now they can act as an acid. And so in their protonated form, their conjugate acid form, they're going to be positively charged. Now, but would you actually find them in that positively charged state? Well, it depends. So basically, we have to look at them. So lysine, arginine, and histidine. Remember that in order to figure out whether something would be more predominantly in the protonated or deprotonated form, we have to compare the pH to the pKa. Or in this case, the pKr, which is just another name for the pKa of a side chain. So lysine and arginine, they have really high pKas. So we're almost always going to be below their pKa, at a pH below their pKa. More protons around, more likely to be protonated. When they're protonated, remember that this is going to be the conjugate acid form. But because it's going to be a really weak acid, it's not going to act as an acid, and so we're basically going to find it in this form predominantly. What about histidine? Well, it actually has a lower pKa. It has a pKa that's actually below where we normally are at. So what's going to happen is we're normally going to be at a pH that's above its pKa. If we're above the pKa, well, now we're going to have less protons around, so you're less likely to find it protonated. You're more likely to find it in this deprotonated conjugate base form. But the pKa is still fairly close to where we're at, so we still have significant amounts of it in the protonated state. But its predominant form at a pH of 7.4 would be its deprotonated state. All of these, however, they're all going to have amine groups that are doing this. And so if you see one of those nitrogens, you're like, oh, basic amino acid. Except if you see a nitrogen like a tryptophan or you see it in an amide, those aren't going to act as bases. But when you see it in one of these three, then it will. And when you see that nitrogen, remember, is almost always colored blue. And so are basic patches, basic amino acids. If you look at them on a protein structure that's like colored by charge, electrostatics, you might see like show surfaces electrostatics. When you show the basic amino acids, you show them as blue. And so you can get like basic patches on a protein, which would be places where they had a lot of histidine, lysine, and arginine. And basically, no pun intended, those are going to be good places for doing things like binding nucleic acids, which they're negatively charged backbone. On the other hand, we have the acidic amino acids. So similarly to how we called the bases because they acted as bases in their neutral form, we call the acidic amino acids acids because they act as acids in their neutral form. So their neutral form, remember, is the carboxylic acid form. Then they deprotonate to form the conjugate base, the carboxylate form. So the two that we're talking about are aspartate and glutamate. And I typically talk about them in that conjugate base form, that tate form, because or ate form, that's going to be the form that we're almost always going to find them in. Because if we look at their pKa, well, their pKa is way below the pH at which we're normally at. So they're going to be almost entirely in their negatively charged form, their conjugate base form. Those conjugate bases, basically, they're going to be really weak bases. And so what's going to happen is we're going to have them be very unlikely to take the proton back. And so we're going to find them in this negatively charged form. So remember, we have aspartate and we have glutamate. They differ in just one. Glutamate's one longer. Both of them are going to have those carboxylic acid groups, and both are going to have those low pKa's. So we're going to find both of them in that negatively charged state. Similarly to how we can show basic things as blue, well, we show these acidic amino acids as like red. And so if you see an acidic patch on a protein, basically it's telling you it's got a lot of aspartate or glutamate. And you can think of the red as being like the oxygen, because both of these are going to have that oxygen. Okay, so other things, like other amino acid things, their polar amino acids are going to have hydroxyl, amine, amide, carboxyl, or thiol groups. And remember, though, that just because tryptophan has nitrogen, we don't call it polar. It's got mostly nonpolar character. We call it hydrophobic and nonpolar. But with like a tyrosine, well, here we've got less of that hydrophobic stuff, and now we have a hydroxyl group. So if you see a hydroxyl group, that's going to be polar. If you see an amide, that's going to be polar. If you see an amine, other than that one, you're going to be polar. And if you see something like charged, like a carboxyl group, that's going to be polar. And you should know, just by seeing the one-letter abbreviation, it's three-letter abbreviation and full name, how to classify it, and what, if any, functional groups it contains. So I don't want you to forget all that knowledge that we just remembered in our amino acid quiz that we rocked. Instead, I want you to that's fair game from now on. So we can all be on the same page when we talk about things with abbreviations. Speaking of being on the same page, we need to be on the same page about what I mean when I say residue. So remember that amino acids are called amino acids because they have an amino group and a carboxylic acid group. But then those groups link together when they form peptide bonds. And so we call what's left over the residues. So what was an amino acid now in the context of a protein is now technically a residue, although we often use these terms interchangeably. Okay, so now let's talk though about that. Those chains aren't just going to stay still, they're going to fold up. So we can talk about proteins as having 4 levels of structure. We have the primary structure. This isn't really like a structure. It's actually just the sequence of amino acids, but it's really, really important. Then we have the secondary structure. This is going to involve hydrogen bonding between the peptide backbone. So this will be between your amine hydrogen, and your carbonyl oxygen. The main forms are going to be your alpha helix and your beta strands. And remember, those beta strands are often going to be connected with beta turns in between to form beta sheets. But the strand is an individual strand. A sheet is multiple strands hydrogen bonded to one another. This is going to give proteins their local structure. Tertiary, this is going to, now we get the side chains of those R group involved. And so this could be interactions between different side chains, interactions between side chain and backbone. And overall, this is going to give a single protein chain its overall shape. But sometimes proteins have multiple chains, and so we can talk about the interactions between those chains using the term quaternary structure. But again, not all proteins are going to have these, and the ones that do, we call these multimers. In a little more detail, let's go one by one, starting with 1, the primary. So remember that this is really just the sequence of amino acids. It's not even thinking about just like a free chain. It's literally just like the sequence on a piece of paper. But it influences the entire protein structure. Because that R group, remember those amino acids, they have all these different properties. The different R group is going to do things like restrict what bond angles can be taken and what secondary structures are preferred. It's going to influence where that part of the structure wants to hang out. So if you have something hydrophobic that's going to be forced into the center of a protein, or do you have something that's hydrophilic that's going to want to be on the surface, hanging out with the water? Do you have something positive and something negative that want to be near one another? So all of these things are going to be influenced by, like, what is your side chain? What is your R group? Which comes down to what is your sequence? And all of this is going to help influence what type of tertiary and quaternary interactions are possible. Because those different groups and the different ability to form different types of intermolecular forces. Okay, but let's step back a second. We go, well, forward from primary, but backwards from tertiary and quaternary. We need to talk about secondary structure, because this is going to be really crucial. So our secondary structure, this is going to involve hydrogen bonding between peptide amine hydrogens and carbonyl oxygens that are going to be, and these are evenly spaced out along the backbone. So no matter what those R groups are, you're going to have this alternating donor acceptor, donor acceptor, donor acceptor throughout the peptide backbone. And all proteins are going to have this. And so what this means is that you're going to kind of get these characteristic secondary structures, things like beta strands and alpha helices. And the reason why you're going to get these is because basically the peptide bond is special. Remember, a peptide bond is just an amide bond. And what's special about it is it's going to be resonance stabilized. It has a partial double bond character. It's involving p orbitals, which are those orbitals that stick up and down. And so you can't rotate. When you have a sigma bond, when you have s orbitals, they're like circular. You can kind of just rotate freely. But what's going to happen with those p orbitals is if you rotate a molecule, well, now they're out of alignment. And so you've broken that bond. And so we want to keep that resonance happy. So we need to keep things in a plane. When we have this plane, basically there's going to be these two places where you can rotate around, and those are on either side of the C alpha. So our alpha carbon, remember, that's the one that's going to be attached to our R group, attached to our side chain. And on either side of that, well, that's where you can actually rotate around. These are called the phi and the psi angles. You don't need to know which is which, but you need to know that the reason why they exist is because this peptide bond is going to be resonance stabilized and thus has to be planar. Now, when you have a lot of amino acids linked up, well, now what's going to happen is basically you can only rotate around each of their C alphas. And so you end up with the chain of planes where you can rotate around specific places. But how much you can rotate is going to depend on things, including the size of the molecules. Because if steric hindrance, basically if you have big bulky things next to each other, even if they're not that big and bulky, if you turn, you're going to run into clashes. The molecules trying to take up the same space, and these molecules, some of the proteins, some of the R chain groups need more elbow room than others. And so this is going to help restrict those phi and psi angles. And then those angles are going to be further fine-tuned to optimize the hydrogen bonding interactions. And this is going to lead to those characteristic secondary string structures like those helices and beta strands or sheets. And so remember that for the hydrogen bonding to be optimal, you have to have like good alignment. And so basically, not only is the movement restricted by steric hindrance, but then you can kind of like fine-tune, restrict the angles further in order to make it so that you have those right orientation for the hydrogen bonds. And all of this together is going to contribute to things like our alpha helices and beta strands that we see show up in all sorts of different proteins. So I don't need you to worry about interpreting any Ramachandran plots or anything like that. But remember that when we were looking at those, basically we saw that there were regions where amino acids were more likely to be, like, remember those were showing our phi and psi angles? And there were various combinations that were more favorable. And there are various combinations within those favorable ones that were specifically found in the different types of strands and sheets. And all that's coming from these contributions of limitations based on where you can rotate, and then those is further limited by the steric strain, and that's further fine-tuned by the requirements for this optimal hydrogen bonding. And you found that different amino acids have a different preference for being in different types of secondary structure. And so basically, if we have an alpha helix, remember when we did our assignment with the alpha helix, we had our styrofoam and then we had our pipe cleaner sticking out of it. Those amino acid side chains are going to be sticking out. And they're going to be like spaced. They're kind of like spread out a little bit on their way down, but there's not that much space, especially close to that backbone before you can kind of get away and get your own elbow room. And so what's going to happen is that the amino acids that can that can form an alpha helix, well, you can have like alanine, which is going to be small and not too intrusive. It doesn't need too much space. But then if you start, you don't want anything bulky, but you can have things that are long and kind of skinny, because then they can kind of like, as long as the bulk's not close to the backbone, like something, one of those like branched ones that has like an isoleucine or something where you've got that big branching near the, where all the crowding is the worst. Or if you have something that's just big and bulky like tryptophan, those want to be very happy in an alpha helix because they don't have room. But if you have something like an lysine, something like an arginine, something that's long and skinny, well, now even though it's like big in terms of how many atoms it has that kind of like bulk spread out in a long skinny form. And once you get far away, there's more room. So in alpha helix, you want long skinny. What about our beta strands? So remember, the beta strands is going to be that individual arrow with that arrow going from N to C terminus. And then you have multiple strands come together and then you get a sheet. And in the sheet, basically you have this pleated sheet where the amino acid side chains are sticking up and down and up and down and up and down, and they're alternating. Because they have plenty of room. So they have more elbow space, more elbow room. And so it's okay to be big and bulky. So if you have a big bulky amino acid, you might find it in a beta strand or a beta sheet, but you're very unlikely to find it in an alpha helix. And because of that alternating nature, basically what you can have is that you can have a sequence where you have an alternating hydrophobic and hydrophilic amino acids that's going to make it so that one side of the sheet can face the aqueous environment and one side can face the interior of a protein. Similarly, in an alpha helix, it's not every other, it's like every 1 and then 4 and plus 4 and stuff. But basically you can end up with one face that is more hydrophilic and one face that is more hydrophobic, making it great for serving as kind of like interfaces between the exterior and the interior environments of a protein. But you can also find that you don't always have to have faces like that. Sometimes you'll find that both sides will be facing an aqueous environment, or both sides will be facing a more hydrophobic environment. It just all depends on where it's placed in the protein. In an alpha helix, the hydrogen bonding that's holding it together, giving it a secondary structure, is within the alpha helix. It's within a single helix. But in the case of beta sheets, well, now the hydrogen bonding is between strands. So there's no hydrogen bonding that's actually taking place between that in the beta strand in terms of when we talk about the secondary structure. Instead, it's when you get these sheets, the hydrogen bonding between the backbones of those sheets those strands is going to give you the beta pleated sheet. And those can be parallel or they can be anti-parallel. Just look to which direction the arrows are facing. And often in the case of the anti-parallel, they're going to be connected by like beta turns. Where in these turns, you're likely to find glycine and proline, but you're very unlikely to find glycine and proline anywhere else. So a glycine or a proline could totally disrupt the secondary structure, could screw up your amino, your alpha helix or your beta sheet. Because glycine, it's too small and flexible, it can loosen up all those structures that you want to be nice and tight. And proline, it's got those weird restricted angles. But those weird restricted angles and like especially like the cis form of it, those can be good for making those tight curves. So you'll see glycine and proline in those curves, in those turns, but not in other types of secondary structure. Okay, so now let's move on from the secondary structure and talk about tertiary structure. So here we have interactions directly involving those R groups. So we saw how with the secondary structure, the R group was going to influence which kind of like structures would form, but not like you weren't directly having those interact. But in the case of tertiary structure, now the R groups are getting involved. And this is going to give the protein chain their overall shape. So the bonds and forces holding them together, these are the same IMFs we talked about before, except that here we have another thing. And so disulfide bonds are cysteine cross links. So basically, when we have cysteine, the amino acid cysteine, and remember it has an EI in its name. So I took off half a point if you spelled it wrong, I'm sorry. But you need to know that the EI form is like the this form, because later when we actually form those cross links, now we can call that cysteine. But EINE is the amino acid form in its reduced form. It's sulfhydryl form, it's thiol form. But what can happen is that cysteines can kind of oxidize and they can form crosslinks where you get the sulfur swapping out a hydrogen for a bond to another sulfur. And this can cause, this can kind of allow for crosslink formation between different regions of a protein. And remember our hair example, our perm, we want those disulfide bonds to form the right way. And so when we break them and then reform them in a perm, that's what we're trying to do. And proteins in our cells, we don't want them forming wrong disulfide bonds, wrong crosslinks. And so we keep an environment in our cells that is relatively reducing. And instead, we only find these disulfide crosslinks typically in like secreted proteins. And they're good in those because they're going to be our strongest type of interaction that we have. And then the rest are going to be our non-covalent. So this is going to be our salt bridges. Remember that in terms of salt bridges, we need the cation, and this is going to be our lysine, arginine, or histidine, our basic amino acids. And then we need an anion. This is going to be one of our acidic amino acids, aspartate or glutamate. Then we have hydrogen bonds. And so remember the ones that can form the hydrogen bonds are going to be our polar amino acids. And then we have other permanent dipole interactions. Again, look for nitrogens and oxygens. And then we have those induced and transient dipoles, our van der Waals, our nonpolar interactions, our hydrophobic interactions, whatever the heck you want to call them. Those would be the weakest. And remember that individually, these are all weak, but they collectively, they're strong. The same types of interactions are going to be involved with quaternary structure, except that here the interactions are going to be taking place between protein chains. So I remember I was always like, I thought a protein was just like a single chain. You know, you've got your the central dogma of molecular biology. You go from a gene to a messenger RNA to a protein. But then you're like, oh yeah, and you can actually have multiple chains join up. And so some proteins are made up of multiple chains. We often say they have multiple subunits. Not all proteins have these, so some act as monomers or single chains. But if they have multiple, we can call them a multimer. We call them a homomer if all the chains are the same, or a heteromer if some are different. And then we can characterize them further based on like if they're a dimer, so 2 subunits, a trimer, 3 subunits, a tetramer, 4 subunits, et cetera. But they're held together by those same types of interactions as hold together the tertiary structure. Okay, so when we talk about protein structure, there's like, we often have ways that we want to refer to various places in the protein. And so if we want to talk about like a part of a protein, we often use this word domain. And so it's a really vague term and we use it in all sorts of different ways. But often it's going to be some sort of largish part that's functionally and or structurally distinct. And so if we look at this example that we looked at before, where we had this protein, it had a kinase domain, which had phosphate groups. It had a phosphatase domain, which removes phosphate groups. And then it has this FHA domain, which allows it to bind to the sites of DNA damage. And so these would be different domains. And you can see that these are kind of like, in addition to being functional domains, it also looks like they're pretty structural domains where maybe even if you were to separate these, they would still be able to fold and act independently. So we often can study protein domains in isolation. These domains are often evolutionarily conserved. And I like to think of them as kind of like rooms in a house. And so just like you might have different rooms in your house that are separated by walls, those would be like structural domains, and different rooms in your house that are not separated by a wall, but you do different things in your dining room and in your living room, even though they're like separated, they're not really separated by a barrier. You can still talk about like different functional domains. Okay, within domains, we often find motifs, and motifs can be found even if we don't have a domain, and like we could have a domain within a domain. It's a really loosey-goosey term, but a motif is typically like a small arrangement of secondary structures that's seen over and over and over again in different proteins, and sometimes has some known function. So this is like our furniture in our room, and so you'll find them a lot of places, just like you'll find a chair in a lot of different houses and a lot of different rooms. But some common motifs you'll find in proteins are things like zinc fingers, which are good for binding to like DNA, Various things like this. You don't need to know examples or anything, but know that it's kind of like a smaller local structure or units made up of typically like some combination of your those base secondary structures. So like your helices and your strands. And then you find these in a lot of different molecules, and even if they're not evolutionarily related. Okay, so when I mentioned like we can study domains in isolation often, well, the reason we can do this is because of recombinant protein expression. So remember, recombinant expression is where we give cells the genetic instructions for making a protein, and they make it for us, or at least we hope they do. Sometimes they don't really cooperate. But in order to give them the instructions, we can recombine the genetic instructions for making them with some sort of a genetic vector. So basically, a vector is like a vehicle for getting something into cells. So typically we stick genetic instructions into like a circular piece of DNA called a plasmid, and then we stick that into bacteria. And then bacteria, we're then going to make the messenger RNA and then translate it to make protein. We also have different vectors that we can use to get things into other cells if our protein is harder to express or make. So one of the benefits of recombinant protein expression, well, as we just mentioned, we could kind of look at domains. So because we're putting the genetic information in, we're telling it what to make, we can alter the instructions for what we want it to make. So we can do things like just study a domain in isolation or introduce other sorts of mutations. We can also, even if we want to study the normal version, we can stick a tag on the end. So we can stick a little extra sequence onto the end, like a his tag or a strep tag, just a few extra amino acids that are going to allow us to do things like purify it, taking advantage of that tag, using resin that's going to bind to that tag specifically. Or we can do things like use antibodies that recognize the tag or various other things. And so we can also put a protease cleavage site in between that tag and our protein. So by putting a few extra instructions in between the instructions for the tag and the instructions for our protein, we can stick the instructions for a protease cleavage site, and then we can actually cut off that tag if we don't want it anymore. So various really cool things that we can do thanks to recombinant expression, including just getting cells to make this, even if they wouldn't normally make it. And we can even like overexpress, so get them to make a lot of it. But there are drawbacks if we were to compare this to just trying to purify the native or like unmodified, naturally made protein. It's going to be less authentic. It might not fold correctly, and it might not have the normal post-translational modifications. So we'll talk more about post-translational modifications later in the course. These are going to be things like glycosylation, so the addition of sugar chains, phosphorylation, the addition of phosphate groups, etc. And so basically, those are going to require specific enzymes that different cells have different versions of, and they might, like in terms of cell types, but insect cells might have different versions of those glycosylators and of those phosphorylators or kinases than like human cells, say, and bacteria might not even have them at all. They also basically, folding often involves the help of chaperone proteins, which kind of help do things like keep those hydrophobic parts from clumping up before the part that they're supposed to hang out with is come out of the ribosome. So basically these chaperone proteins are going to help things fold, but different cells will have different types. Cells might not want to make your protein, or making a lot of it might be toxic to them. So yeah, you can tell them to make a lot, but if making a lot of it kills them, then that's not going to work well. So what you might have to do is try multiple cell types to best mimic the real deal. Bacteria, this is our cheapest and our easiest. They're good for small, easy proteins. But if you need something that you're dealing with something more complicated, something that's normally in a eukaryotic cell, so not bacteria, you might want to try doing another type of eukaryotic cell. So insect cells, more human-like, but they might have slightly different modifications, but they're easier to scale up and cheaper than mammalian cells, which are the most human-like, but they're the highest maintenance and most expensive. In order to purify proteins, we often use protein chromatography. So this is the main method of protein purification, and basically what we do is we take solutions that contain proteins and we flow them through columns filled with little resin. And this resin has different properties. Different proteins will interact with the resin differently, and thus you can separate the proteins. Oh, and just a word that when something is like coming out of the column, we say that it elutes. So there are three main types of protein chromatography, and you don't need to know like all the details. You don't need to know specific column names or that sort of thing. But you should know that there are these three main forms: affinity chromatography, ion exchange chromatography, and size exclusion chromatography, which we also refer to as gel filtration. So with affinity chromatography, your protein is going to bind based on some specific feature, such as having a his tag that complements the nickel on the nickel NTA resin, or strep tag that will bind to the streptactin resin. So you have some unique feature, and this could be an affinity tag, and it often is, but it could also just be some natural property of the protein, such as using like a lectin column if you have a protein that's glycosylated. There's also ion exchange chromatography. This is going to separate based on charge. So proteins stick to oppositely charged resin, then you can peel them off with salt. And then you have size exclusion chromatography, which is also known as gel filtration. This is going to separate proteins based on their size. So the name kind of gives it away in that case. But basically you have the beads are, instead of interacting, like sticking to the beads, things actually go through the beads and around the beads. And their beads are basically porous. They're like gel. It's like a gel. But they're in this little bead form. And the pores are different sizes. And basically the bigger proteins can't go through all the little tunnels. So they get to take shortcuts, and so they travel faster. And then the little guys have to go through a bunch of tunnels, so they travel slower. But all you need to know for this, the context now, is basically that size exclusion chromatography is going to separate things based on size. Depending on how pure you need something, you might have to use multiple sequential columns. So when we were looking at our protein and our structural biology papers, we were seeing that they were doing often doing a lot of columns like in a row. And so, yeah, you often do that with structural biology, as I know very well, coming from a structural biology lab where I did my PhD. And so when you are doing multiple columns, and even if you only do a single one, you're often going to want to choose affinity chromatography. This is going to be the most specific because those other proteins in the cell, they're not going to have a strep tag, but your protein will. But there are other proteins in the mixture that are going to have charges PIs near yours, and other ones that are going to have sizes near yours. But the affinity chromatography, that's going to take advantage of the most unique thing. It's going to be the most specific and let you get rid of most other stuff. Ion exchange chromatography. Now this you can use no matter what your protein is. It's going to take advantage of charge because like all proteins are going to have a PI, right? They're just going to have different PIs. So remember the importance of the PI was that proteins become more positive when are positively charged below their PI, and they're going to be negatively charged above their PI. And so this is like that real world application when we're talking about that PI in the very beginning. And so basically with cation exchange chromatography, you want to use this for your basic proteins. Remember those basic proteins have a high PI. They have lots of those lysine, arginine, histidine. And we want to use at a pH below the PI so that it is positively charged. We're exchanging out the positively charged cationic protein for the positively charged cation in salt. So we can get our protein to stick to negatively charged resin. Then we let everything else flow through. We start increasing the salt concentration, and the cation in the salt is going to compete off our protein. We're going to exchange our cationic protein for the cation of the salt. So this is cation exchange chromatography. If, however, our protein has a low PI, we say it's acidic, it's got lots of glutamate and aspartate, well, then we're going to want to use anion exchange chromatography. Our protein is typically negatively charged because we're typically at a pH above the PI. And so our protein is going to be negatively charged, and we're going to want to use anion exchange chromatography, where basically now we're sticking to positively charged resin, and we're exchanging our anionic protein for the anion of the salt. You can do this in like gradient or in step, but the same idea each case. And then finally, we saw from the spike papers that memory proteins are challenging to work with, but it is possible and they can be extracted with detergents. So you don't need to do any calculations on this exam, and there will be more in binding affinity in the upcoming exam, the next exam, especially incorporating our things on cooperativity and allostery. But I do want you to be able to kind of connect the binding affinity to our concepts about intermolecular forces to be able to say, okay, well, if this can form more bonds, if we can form a stronger bond, well, now we're going to have a higher binding affinity, and so we'd have a lower KD, which is equal to, we can write KD in all these different forms, and I want you to be able to kind of like understand what this is saying. But the key focus is to focus on the concepts. So basically, the KD is the main way we talk about and compare binding affinities, but it's the equilibrium dissociation constant. So the KD is going to be equal to, if you have two binding partners, A and B, the concentration at which the one partner at which you find half of the other partner be in the bound state is going to be your KD. So mathematically, it's equal to the concentration of your A times your B over the concentration of your bound AB. And the reason why we use this instead of the equilibrium association constant Ka is because we have units of concentration, so it's convenient. Now, it's going to equal mathematically, it's going to be like that like law of mass action thing where we could write some sort of like equilibrium equation for any sort of reaction. Well, here we're talking about the equilibrium equation for going in the reverse direction. We're unbinding of molecules. And so if we have two molecules, A and B, we mix them together, we let them reach equilibrium, then we can basically write an equilibrium equation, and then the KD, so our equilibrium constant, which in this case is our equilibrium dissociation constant, is going to be the concentration of A times the concentration of B over the concentration of the complex AB. So our unbound over our bound. It's going to have, the reason why we use this instead of the Ka, which we'll discuss in a second, but the association constant, is that it has units of concentration, which is more convenient. What's really convenient is that the Kd is going to be the concentration of one binding partner at which half of the other is bound. And remember, we can talk about the amount that's bound is like the maximum amount bound would be our Bmax. And the half of that would be our half Bmax. At a concentration greater than the KD, more than half is bound, and at a concentration below the KD, less than half is bound. So if there's less around, less of it's going to be bound. If there's more around, more that's going to be bound. So again, we have that sort of K number, a number with the K that's telling us about a halfway point. The lower the KD, the stronger the affinity. So because we're dealing with the reverse direction, if we have a bigger KD, that's going to be weaker. If we have a lower KD, that's going to be stronger. And it's easiest to think about this in terms of the concentrations, because basically if something has a stronger affinity, you need less of those chances to bump into the thing in order for you to find half of the copies bound at equilibrium. Now, this could be because you bind tighter, or it could be because you release faster, or it could be some combination of the two. So this gets us to the other way we can represent the KD, which is, so we see those little Ks, not the capital ones, but the little italic ones, those are telling you about rate constants. So this would be like our K on, our on rate, and our K off, our off rate. And the KD is going to come from our K off over our K on. So KD is an equilibrium constant. It doesn't tell you anything about the rates. If you want to know about those rates, you have to directly measure them, or at least measure one of them, and then also measure the KD. So if you have two of them, you can find the other one. But basically, the KD is just going to be an equilibrium constant. Okay, what about our Ka? Well, this is the opposite. This is the equilibrium constant. It has units of 1 over concentration, which isn't convenient, which is why we use the KD, which is 1 over the Ka, which has units of molarity or whatever your concentration unit is. So the Ka, though, has a direct relationship with affinity, so the higher the Ka, the stronger the affinity. You should be able to interpret and sketch binding curves, both linear and semi-log scale. Remember that all of your binding curves are going to look sigmoidal on a semi-log plot, even if they don't show cooperative binding. In either case, basically the higher affinity things will be shifted to the left. It's going to take a higher concentration of partner to reach half of the Bmax. And remember, the significance of half Bmax is that's the concentration where you find that is going to be equal to your KD. Lower affinity things are going to be shifted to the right, telling you it takes less of the partner to reach half Bmax. And so you should be able to kind of like sketch out curves. If I say like, well, if you have two things, one is a higher KD than the other, or one has a lower affinity than the other, you should be able to kind of draw a couple curves, just estimating things out. Also be able to recognize where we have Bmax, half Bmax, or KD. Finally, try not to stress. My goal is for you to learn. This exam isn't worth a lot of your grade, but it's an important way for both me and you to assess that learning that you're doing, and in a way to make sure that you're not just trying to memorize things. It also serves as a great push for you to review this foundational biochemistry content. So I'm not just wanting you to learn this so that you could, just for the sake of learning it, but also so you can see how it applies all around us. So when we're doing our like biochemistry all around us discussions, when you guys are telling me all those cool examples you're finding of how biochemistry is coming into play in your life, when we're doing those readings of those articles, dealing with real world situations, biochemistry is life, biochemistry is all around us. And by learning this stuff, we're getting such, like, it's real privilege. Learning is a privilege, and getting insight into this molecular world, I hope that you don't take it for granted. I hope that you try to learn it, and I hope that you see how exciting it can be. So I wish you all the best of luck on your exam, and I want you all to do great. If most people aren't doing great, then that's my fault, not yours, and I won't punish you. And I'll instead, I'll adjust things. But your grade is not on a curve or anything, so the grade of the person next to you, the grade of your friend, doesn't impact your grade. I want to give you all A's, but you guys have to work for that too. And so if you have any questions, reach out, let me know. And good luck. Try not to stress. We're all in this together, and this really is just to help you learn.